Have you ever wondered if microorganisms can get sick and perform less actively in biotech processes? A process I was interested in my PhD is the anaerobic digestion process, which is mostly performed by bacteria and archaea, which are degrading organic material in the absence of oxygen to biogas, what we then can use, for example, as a renewable energy in anaerobic digester plants, as you can see here on the picture. But when we are talking about cells, we also need to talk about viruses, because everywhere where you have cells, you're also expecting viruses. And phages are viruses infecting bacteria and archaea. And when we look into the fermenter, at the moment, their role is completely unclear. Maybe they make our working microbes for the process sick so that we have less biogas, or on the other side, they're lysing cells that are not necessary for the process and releasing nutrients. Or else alternatively, maybe they are the process police and holding the balance between the microbes in general. One method to study microbial communities is metagenomic-centric metaproteomics, where you're studying the proteins of sequenced genomes of your samples. But the problem is when we are talking about proteomics, we are talking about biomass. And when you look here, viruses have a very low biomass, so they are really hard to detect. And at the moment, there are a lack of workflows that you have a chance to understand their impact on such processes. And in my thesis, I developed workflows that you have a chance to reliably detect phages on the one side, and on the other side that you can study newly synthesized proteins because they give you the direct response of your microbial community. When we are talking about newly synthesized proteins, we are talking about metabolic labeling. And one method is BONCAT, where you label your newly synthesized proteins, but also your phages. And the advantage to other methods is that you can combine this with click chemistry, so that you can modify your proteins, but also your phages. And once you have labeled phages, I developed a workflow where you can attach fluorescent dyes or biotin to modify your phages and do, for example, host screenings or some purification of the viruses. And in general, when we are talking about anaerobic digesters, we're talking about very difficult sample types. And this means also a workflow was necessary to enrich phages from such systems. And I designed this workflow also that you have the chance to use click chemistry. In our test with mass spectrometry and metagenomics, we found 3-fold more phage genomes and 6-fold more phage proteins in with this workflow. And once you have labeled communities, also the proteins of the community is very crucial, very valuable. And I developed also a workflow here that you can enrich such proteins from complex sample types to get information about the microbial adaptation and interactions. So that we can, in summary, say that we now have a toolbox that we can study phages in complex systems, which we can adapt to also to other systems, and it's very flexible. So that we can now say in the end that we can investigate if microorganisms can get sick and perform less actively.